Hi everyone, in this video I'm going to introduce you what is biodegradation and how does biodegradation relate with microbial physiology. Biodegradation is the future of the world to solve pollution. As the term biodegradation shows, it is a process which is conducted by microorganisms to degrade organic materials. In a formal definition, it was meant by transforming an organic substance through biochemical reactions by the action of microorganisms. So, why is biodegradation able to solve the pollution issues that are widespread in the world? Some hazardous toxic contaminants can become the nutrients for some microorganisms, and microorganisms will further work on breaking down the organic metals. By benefiting the microorganisms with the contaminants, reduction of waste and cleanup of environmental contaminants can be achieved. However, not all organic molecules are able to be eaten up completely. It is still depending on the nature of microorganisms. Generally, there are three different kinds of biodegradation. The first type is the modification of the chemical structure of a substance to disrupt their specific property. The second type is the biodegradation of a substances by using microorganisms to remove undesirable properties of a compound, and this is normally more environmentally acceptable. Third, a complete breakdown of a compound either fully oxidized or reduced to simple molecules. Some researchers have shown that the third type of biodegradation always produce more harmful products than the substances degraded.
Biodegradation is a redox process and it can occur without much restrictions as bacteria has a high survivability and are able to adapt to its environment easily. However, for successful biodegradation to take place, there are requirements that are to be met. Firstly, the microorganisms will require its catabolic potential. The microorganisms must contain the genes needed for coding the required enzymes so that the biodegradation can take place. Secondly, the presence of microorganisms at the degrading site is crucial. The compound must be rid of its chemical and physical barriers so that the microorganisms can act upon it. Thirdly, a carbon source is needed for the cell growth. Nutrients including nitrogen, phosphorus, calcium, magnesium, iron, and trace elements are also important, with nitrogen and phosphorus needed in the largest amount. Moreover, the substrate will have a higher rate of being degraded if the substrates are suspended in an aqueous solution. This is to provide a medium, thus allowing the transfer of electrons. Furthermore, the microorganisms are required to have inducible enzymes, and its presence will lead to the lysis of substrates. This is because the enzymes are not always produced due to the reason of saving energy for other purposes. Besides that, there are subconditions that are needed for biodegradation to take place smoothly, as the presence of oxygen, depending whether it's an aerobic or anaerobic microbial reaction, adequate temperature and pH level, absence of substance toxic to the microorganisms such as heavy metals, absence of easily degradable substances. This is because a preference will establish to break down the readily ones rather than the non-readily ones. In conclusion, the microbes will with the required catabolic capacity must be present in sufficient numbers to exert measurable degradation rates. Environmental and thermodynamical conditions must be favorable to sustain the expression of this capacity.
Petroleum-based products are the major source of energy for our daily life and industry. However, oil spillage has always been a troublesome issue that intensifies the problem of world pollution. Those oil spillages always contain some toxic hydrocarbon that will threaten the environment and marine ecosystem. While hydrocarbon is too complicated and financial burden to be detoxified by using conventional technique. Therefore, the idea of bioremediation was proposed to be the solution. Bioremediation is a process of detoxifying oil spillage based on biodegradation, and most importantly, it is cost effective and environmentally friendly. In this topic, I'm going to introduce you the general mechanism of degrading hydrocarbon by microorganisms. Generally, the degradation of the majority of organic pollutants is optimum under aerobic condition. This figure has shown the main principle of aerobic degradation of hydrocarbons. There are 9 bacterial strains have been found to have the ability to degrade crude oil. For example, Pseudomonas fluorescens, Pseudomonas aeruginosa, and Bacillus subtilis. As hydrocarbons act as the nutrient source of microorganisms, hydrocarbon has to be administered into the intracellular of microorganisms to undergo further degradation. After that, pollutants will be attacked by oxygenase along with the incorporation of oxygen. Subsequently, the products will be converted step by step into one of the intermediates that were needed by a specific metabolic pathway for central intermediary metabolism. For example, tricarboxylic acid cycle. After a few rounds of the metabolic pathway, biosynthesis of the cell biomass can finally be synthesized from central precursor metabolites like acetyl-CoA, succinate, and pyruvate, and they are useful for the growth of microorganisms. In the meantime of feeding hydrocarbon to microorganisms, the pollutants are eliminated owing to the degradation of microorganisms.
The degradation of plastics are caused by microorganisms such as bacteria, fungi, and actinomyces. In order to use such materials as a carbon and energy source, microorganisms have developed a special strategy and is explained in this flowchart. Number 1. Attachment of the microorganisms to the surface of the polymer. Number 2. Growth of the microorganisms using the polymer as a carbon source. Number 3. Ultimate degradation of the polymer. As long as the surface of the polymer is hydrophilic, the microorganisms will be able to attach to it and perform biodegradation, causing the lysis of the polymer into oligomers, dimers, or monomers, thus allowing the microorganisms to obtain energy source and proliferate. The lysis of the polymer follows a biochemical pathway which is explained as follows. Firstly, biodeterioration takes place. It can be defined as the action of microorganisms and decomposers causing degradation that changes the mechanical, physical, and chemical properties of the plastic. In most cases, abiotic parameters contribute to weaken the polymeric structure. The microbial film provokes serious physical and chemical deterioration. Physical deterioration is when the microbes that forms the biofilm secretes extracellular polymeric substances, EPS, that reinforce the cohesion of the biofilm and the adhesion to the plastic surface. The EPS enters the pores. Microorganisms can then grow inside, thus increasing the pore size and provoking cracks that weaken the physical properties of the plastics. Chemical deterioration occurs when the microbes that develop the biofilm releases acidic compounds such as nitrous, nitric, sulfuric acids, oxalic, citric, and fumaric acids. The pH inside the pores is then decreased, resulting in a progressive degradation that changes the microstructure of the plastic matrix. Secondly, biofragmentation takes place, which refers to the catalytic actions that cleaves polymeric plastics into oligomers, dimers, or monomers by enzymes secreted by the microbes. Since plastic polymers are molecules that are too large to cross the cell wall, it needs to be fragmented into smaller molecules so that it can be used as an energy source. However, plastic polymers are very stable due to long chains of hydrocarbons with balanced charges, hence the microbes will require enzymes such as oxygenase, which can add oxygen into a long carbon chain. This results to alcohol or peroxyl groups, which are more susceptible to biodegradation. Other transformations are then catalyzed by lipases and esterases after the formation of carboxylic groups or by endopeptidases for amide groups. Thirdly, assimilation characterizes to the integration of molecules transported in the cytoplasm in the microbial metabolism. The assimilation results in numerous secondary metabolites that can be transported outside the microorganism that do not have the metabolic capability to transform it or do not need to metabolize or to store it. The secondary metabolite excreta may be used by another cell that can perform further degradation or stay further in the pool of non-assimilable compounds. Inside the cell, the plastic monomers are oxidized through catabolic pathways to produce energy, cell structure, and new biomass. Finally, mineralization refers to the complete degradation of molecules that resulted in the excretion of completely oxidized metabolites. The mineralization refers to the complete degradation of primary and secondary metabolites that resulted in the excretion of completely oxidized metabolites.
Among biological agents, microbial enzymes are one of the most powerful tools for the biodegradation of plastics. Several factors may be explored for a better biodegradation of polymer plastics. By using surface active agents or inducing the microorganisms to produce surfactant to allow better attachment of microorganisms on the polymer surface, by blending the polymers with biodegradable synthetic polymers such as polylactic acid or polycaprolactone, by the pretreatment of the plastics, which includes thermal, UV, high energy radiation, and chemical treatment, bioaddition by culturing those microorganisms that can efficiently degrade the plastics, and biostimulation by addition of nutrients that may be limiting in the environment. There's a huge demand in exploring these microbes which can grow in different conditions and under specific stress conditions, may be directed to grow and use the plastic carbon polymers as energy source. Recently, a novel bacterial strain called Ideonella sakiensis was discovered that proficient in degrading PET by using it as a carbon source and energy. PET is a non-degradable fossil-based polymer. Additionally, other studies have shown that the microbial combination composed of Pentoea and Enterobacter has the potential to degrade the LDPE. Other than that, scientists also found that some microbes are able to convert organic styrene, a waste material of plastic, processing into PHA, which is polyhydroxy alkanoates. Pseudomonas putida NBUS12 is the microbial strand that is efficient in degrading styrene. Achromobacter psilocybins has recently been found to affect the structure of high-density polyethylene. All of the research efforts in the above have shown that biodegradation is a feasible and applicable technique to solve the pollution issue. The world that we are living right now has been challenged by the infinite number of ways that will lead to pollution, for example, plastic. Therefore, instead of using the conventional plastic bag, we could start using the biodegradable plastic bag in the future, as that could become the food source of microorganisms.